Iron. This is going to be part 2. We're going to look at iron and the method of uptake and transport across membranes. In plants, transport across the plasma membrane is closely linked to iron 3 reduction, which means we're going to go from iron 3 to iron 2. So let's look at how this goes. We start out with a membrane, and this is our standard lipid bilayer. This is a cross-section of it, and we're going to start out saying that the left-hand side is the apoplast, the right-hand side is the symplast. Remember, apoplast means outside of the membrane. Symplast is inside of the membrane. So symplast is inside of the cell. We have an enzyme, a protein that goes across the membrane, which in this particular case is represented by this long bar that goes across. That is called iron reductase. There is also a channel, which is an Fe channel, which is where the iron is going to move through. We start out with an NADPH forming NADP and releasing a hydrogen ion. This causes 2 electrons to be released on the apoplast side of the membrane. These 2 electrons then combine with 2 iron dicitrates. Notice it says dicitrate, which means it's got 2 citrates in it and is going to release 4 citrates and the iron. The 2 irons are going to be modified from iron 3 to iron 2. The iron is then able to go through the Fe channel through the membrane. Now it's on the inside of the cell. When we're looking at transport across the membrane, it's a rather simple process, but notice it's aided by this particular enzyme, Fe3 reductase, which requires energy that we get from NADPH. Therefore, this is an active transport. It is not a passive transport, is it? It is an active transport across the membrane. When we talk about uptake of iron into plants, we've got uptake in grasses, we have got uptake in other plants. In grasses, it occurs with non-protein amino acids. Now, what that means is here we've got 2 non-protein amino acids. We can see that they are amino acids because they've got the amine groups in there. We can see that they've got the acid groups, which are the CO2 minus. And in this case, we're looking at mugenic acid and avenic acid. These look very much like a fatty acid, but they are a little bit different than the fatty acid because they've got the amine group. So these are chemicals that are secreted by certain plants. They are released by the root tip of grasses. They will complex onto iron and then they are absorbed. They are released in the greatest quantities in the morning. So you have the plant where it will actually have these released in the morning, which will help bring the iron in so that you've got sufficient iron during the day. The uptake is independent of soil pH. Remember, we said that iron is more available in acidic pH and less available in higher pHs. Well, here we're trying to get the pH out of the equation by having these materials which can bind onto iron and bring it into the plant. The other thing that is interesting is these are rapidly degraded by microbes, which means they don't stay around in the soil. When the soil microbes start getting up, when the temperature warms up a little bit, they will degrade these and they will break these down. When we look at iron, we see the way we get transport across the membrane. We also see the way that we get uptake in grasses using specialized types of materials, which are non-protein amino acids.